All right. Well, thank you all for coming today to today's Grounds on the Go seminar. This is a series that my office, the University of Virginia's Applied Research Institute, and the UVA Research Park put on together, where we try to bring the best researchers and scientists and intellectuals from the University of Virginia up here to the Research Park to engage with the community and the tenants of the park. So we will be having this series again starting in the fall semester. So now that we have your email list, be on the lookout for new series, new seminars in the fall semester. But today we have an excellent talk about human genome editing and the role of CRISPR. And so I'd like to introduce our speaker today. Mazhar Adli is an associate professor in the Department of Biochemistry and Molecular Genetics at the University of Virginia School of Medicine. His lab's ultimate research goals are to understand the key drivers of cancer and identify novel therapeutic drug combinations to prevent cancer development and chemotherapy resistance. To achieve these goals, the lab applies genomic and epigenomic mapping, editing, and imaging approaches to understand genome regulation in normal and malignant settings. Professor Adli earned his PhD at the University of North Carolina at Chapel Hill, and he did his postdoctoral research at Harvard Medical School and the Broad Institute of MIT and Harvard. He is an associate member in both the Center for Public Health Genomics and the Emily Court Cancer Center, and in 2019 he was named a Penn Scholar in the School of Medicine at UVA for his scientific experience and expertise in productivity, mentorship, and service to the greater research community. It's a great honor to have you here today, and I'm excited to hear your talk, so thank you so much. Please welcome.
Thank you so much. Correct the typo. This is Mike on. I'm happy to be here and discussing the CRISPR technology. I want to put the CRISPR in a bigger picture and how this technology is now empowering us to do things that we have never been able to do before. For billions of years, the life on Earth was driven by two main forces. There was a random mutations in DNA and a natural selections of this DNA that drive the life on Earth. Around 10,000 years ago, we as human species, we changed the course of life on Earth. Now, for the first time, we were still, the mutations were random, but we carried out the non-natural selections. So through selective breeding, we changed the course of many species around us. And now, right now, around 90% of the species that are living around us are not naturally selected. So we humans decided what's going to live and what's going to die on big part of this world. But now we are entering a new age of this evolution. Now we as human species, for example, the example of this non-natural selection, this is the wild type corn, and we non-naturally selected, and now we have this corn. This is not, I'm not talking about the genetically modified version. This is the corn that normally is grown. Similarly, this is a wild type banana, the original banana, but this is a non-naturally selected version of it that we decided, we selectively breeded this version, and now we have this one. So we have been pretty successful in deciding, what's going to live, what's going to survive. And this is one of the successful cases. But for some other things, we were not as successful, maybe. But the dog species, all 100% of the dog species around us, they are all non-naturally selected. So they are not wild-type species. But now we are entering a completely new era with the CRISPR technology or with these genome editing technologies. Now, for the first time, we are making non-random mutations in the DNA, and we are non-naturally selecting these mutations. So this empowers us to do completely things that we have never been able to do before. And this allows us to do genome editing or DNA editing. So I want to briefly talk about what is DNA, what is genome. So the life code of all living things on Earth is DNA. And the DNA from every organism looks essentially the same. So there is no difference, and it's composed of these four letters T, A, C, G, and T pairs with A, C pairs with C. This is an example of human genome. It contains 3.2 billion of letters of A, G, C, T. And it's literally 2-meter-long molecule that fits into the tiny nucleus of every cell in our body. And we have trillions of these cells in our body. So each cell contains this much information. If you try to print this one out, it will basically make a 200,000 page of kind of phone books. This is how much information is included in each of our cells. And as I said, we have trillions of these cells. So you can imagine how much information we carry around with us. It's the largest library that has ever been created for a library this size has never existed. When we over the years, of course, we developed technologies that allowed us to read this DNA, and you can read this whole thing, but it's pretty boring. This is, for example, a genetic code for an insulin gene. So this is a human insulin that regulates our glucose level in our body. This is the information that codes for it. If you take this information and put it into bacteria, bacteria produces exactly the same insulin. So, and that's how the recombinant insulin is currently used as a medicine. It's basically we are enforcing bacteria to produce basically produce a protein that is coded by our own gene. So the same DNA functions exactly the same way it functions in bacteria, to human, to yeast, or to plants. Is exactly the same genetic information. But this genetic information was a read-only file to us for a very long time. We could take this DNA, we could isolate it, we could read this DNA, but we were never able to change this DNA in living things. But now with the genome editing technologies, this is changing now. So this genome editing technologies now allows us to not only read this DNA, but also to write it, to rewrite it, to edit it, to change it. And this, of course, is what gives us this amazing power or to non-randomly add mutations or add specific sequences to DNA. So CRISPR is, of course, the latest version of this technology, but there were technologies before CRISPR as well. So since 1990s, people were trying to develop new kind of proteins that will go and bind to a specific part of the DNA and will make a double-strand breaks in the DNA. So meganucleases, there were zinc finger nucleases, talons, but all these guys were protein-based. So you had to re-engineer proteins, and only very, very specialized labs could do it. But with the CRISPR, we don't need to re-engineer a protein, because the protein part of the CRISPR is the same. It's called Cas9, or there are new versions. But the things that allows us to send or to direct this enzyme is an RNA. And with this one now, we can send this to a specific region in the genome that we decide, and it will make a double-strand break at these sites. This is actually all that CRISPR or all the genome editing technologies do. So they just make a break in DNA. Now you can imagine, why would we do that? Why would we intentionally send this to somewhere in the genome and make a break in the genome? Because DNA is the most essential molecule for any living things. You don't want to break it. And when you break it, cell has to repair it. And there are two ways cells or living things repair their DNA when there is a double-strand break. One is called this, non-homology end joining. Basically, this one is a very quick kind of fusion of these two ends of this DNA molecule. And when this quick fusion happens, then there are some mutations, random mutations accumulating at those break sites. The alternative way is cell can also use a template to repair it. This is a more precise way of repairing it. So in this case, you can disrupt the gene. And in this case, you can precisely edit DNA. And this allows us to correct genetic mutations, for example. This allows us to maybe delete certain genes that we don't want. So this is a kind of a summary of the CRISPR system. So if you could keep in mind these blue words, then you can walk around as an expert on CRISPR and you can show off. So it's a bacterial immune response. This is the system that has been evolved over billions of years because of this arms race between bacteria and viruses. Viruses try to infect bacteria, and bacteria try to save themselves. So bacteria develop this system called CRISPR, so that when virus infects the bacteria, they take this DNA and put this DNA into their own genome as if carrying a copy of the picture of your enemy. And whenever that same virus infects again, now the bacteria has an ability to recognize it and destroy its DNA. But scientists figured that it's, in 2012, they repurposed this as a genome editing tool. The system is composed of two components. One is called Cas9 protein, and the other one is a short guide RNA. So this is what the thing that cuts DNA, and this is that guides this Cas9 to anywhere we want. And the beauty of it is that we can custom design this guide RNA. So that we can easily, within a few hours, or if you are in the lab in a 24-hour, 48-hour, we can design a new guide RNA and now send this to a completely new region in the genome. So at the target site, it binds to the DNA, and because of this enzymatic activity of this Cas9, it will make double strand break. And as I showed in previous slide, when double strand breaks happen, you can make specific changes in DNA. But we can also change things. We can we can change a little bit of this Cas9 by changing two residues of it. Now it no longer cut DNA. So now it can only bind to DNA. So now this one can be repurposed to do many other things. Because now this provides as a molecular shuttle to send this thing with some payloads to a specific region in the genome. For example, we can fuse activators to this dCas9, which is the D stands for dead Cas9, or the Cas9 that doesn't cut DNA. We can add specific proteins that will activate genes, or we can add repressors that will repress the genes. Now we can specifically, among thousands and thousands of genes, we can send this molecular shuttle into the nucleus, and we can turn genes on and off. And this was, until 5 years ago, this was a holy grail in molecular biology, for example, to be able to send something into the nucleus to a specific gene and shut it off, or send to another one and turn it on. And this happens without changing DNA. This is in addition to wild-type Cas9. So in our lab, we are interested in genome regulation. How is this, our genome, these 3.2 billions of letters, how is this regulated in normal cells, and how this regulation go wrong in disease settings, and how we can understand this process and manipulate it, hopefully, to correct disease state back into the normal cellular states. So when we talk about genome regulation, there are, at the big picture, there are at least 3 layers of regulation. One is the DNA sequence information. Obviously, if there is a change in DNA sequence information, things go wrong from there on. But even if the DNA sequence is perfectly fine, this DNA sequence is regulated at the chromatin structure, what we say chromatin. Because DNA in the living things, inside our cells, it's never alone. It's always associated with this specific set of protein that packages this DNA in the nucleus of each cell. And anything that aberrant regulation in this packaging step can also determine whether that gene is going to be turned on or off in this particular settings. And there is third layer of regulations, 3D organization is also important. It dictates whether a gene, if it's in the center of the nucleus, that genes tend to be highly active, or if it's on the periphery of this nuclear periphery, then those genes are silenced. And we still don't understand quite well how this 3D organization of the genome is regulated in normal and disease settings. And the reason we don't understand is because we don't have necessary tools to study these kind of things. Now CRISPR allows us to study all these different layers of regulation in the genome. And I will give some specific examples. As we were studying this thing, we, of course, since the last 4 or 5 years, now we can use CRISPR to do gene editing. We can do base editing. I'll talk about this a little bit. We can do epigenetic editing. We can bring this different piece of DNA together and how they study how they regulate each other, and do this live cell chromatin imaging that I will briefly mention. So early on, it was wild-type Cas9 and this dead Cas9. And people, researchers, keeps adding new function to this tool. And now it's becoming really a multifunctional tool that allows us to do many other things. And this thing is apparently you can buy this. It's I don't know how you can use it, but. So one of the main questions about CRISPR was, how specific is this? We found this amazing tool now. You can send it to somewhere and make necessary changes, but does it do anything else across the genome? This is basically a $1 million question for CRISPR technology. And it's very difficult to study this effect because our genome is large, right? I show this library, there's 3.2 billions of letters of information, and we can send CRISPR by picking a 20 nucleotide sequence. For example, if you are sending this sequence, let's say, is an effect versus effect, is it going to go and change those other words in the genome as well or not? So when I started my lab, one of the first papers from our lab was to use this technology we call chipstick technology, which allows us to study where a certain protein binds to DNA across the genome, across this 3.2 billions of letters of information, where a DNA binding protein would go. And this technology allows us to map those binding sites for this protein. And we thought we can use this to study how Cas9 binds to the DNA. And when we found this one, we discovered that if this is the sequence that you target DNA, around this first part of the sequence, it never allows mismatches, which means that this part needs to be perfect. But at this distal part, even when it's not a perfect match, it still binds to it, which means that CRISPR has some off-target effects. So it's not super specific. So I should add that since we published this one, the field is evolving quite amazing at an amazing pace. Now there are higher fidelity version of CRISPR-Cas9 that will have less off-target effects. But we should realize that it's not a perfect tool. But then we also we designed some computational algorithms to predict where are these target sites in the genome. Because if we know before we do an experiment, with the in silico analysis, they said that if we target this sequence, these are the potential off-target sites that it may edit. Then when we do actual experiments, we can go to those sites and study them, whether is it actually binding or is it actually editing or not. We can do epigenetic editing. We could do design, instead of adding some activators or repressor, we can also add fluorescent protein to this thing. And now we can use this molecular shuttle to image DNA, image our own favorite gene, and see how it is regulated in this three-dimensional space in the nuclear space, and how it moves, and we can study those kind of dynamics as well. Now the wild-type Cas9 or this all other genome editing tools, as I mentioned, they go through this double-strand break stage. So that's how they work. And because of these double-strand breaks, then we can make whatever changes we want. But if there are too many breaks in the DNA, for example, because of if this guy binds to many places in the genome, it creates many double-strand breaks. And when this happens, cell thinks that something catastrophic is going on, and it undergoes apoptosis. So it undergoes cell death, basically. Because this is like a higher level of radiation on the cell, that it shatters the DNA, and cell is dying because of this thing. So this base editor, CRISPR base editor, is a new version of CRISPR technology. Now it converts one base specifically into another base without going through this double-strand break. So cytosine, for example, is one of the bases in DNA. This, as you can see, it's based on a CRISPR, but scientists add new proteins to it. And because of these new proteins, we can convert cytosine directly into thymine. This is another base in the DNA. This doesn't go through the double-strand breaks, which means that there is no cell damage or there is no cell death. And we repurposed this to create a new version of CRISPR where we can efficiently silence genes, for example. And new versions also have been produced now that converts A into G as well. So this goes through convert one base into another one. It's an amazing, because a lot of genetic diseases, which there are 6 thousands of these genetic diseases is because of the single point mutations. And that a single point mutation can create a disease. And that's what now these tools are becoming very favorable for this kind of treatments. Another thing that we use CRISPR in the lab is to screen genome to find out what are the novel tumor suppressors and or the new drug targets in cancer. So this is an amazing power for us and for the field to be able to look in the genome and screen entire genes in 20,000 genes to say which one of these genes is important for cancer formation or tumor formation, and which one of these genes can we target, can we delete, or can we inhibit so that we can make a drug work much better way. And this is one of the areas of the research that we do. And we apply this in pancreatic cancer because it's one of the deadliest diseases, cancers that is around. So briefly, what is pancreas? So pancreas is composed of two glands and fused into one single organ in our body. One of the glands is secreting this insulin and glucagon. This is what regulates our blood sugar level, basically. But there's other cells in the pancreas. These are secreting this enzyme that helps us to digest our food. So the pancreas is basically composed of these two types of cells and these two types of glands. And large majority of the pancreas is composed of these exocrine cells that secretes this enzyme. And this is where the pancreatic cancer, vast majority of pancreatic cancer arises from these ductal cells. Around the world, more than 300,000 new cases. As you can see, this is the number of deaths is almost equal to the number of new cases. It's because 80% of people die within the first year once they are diagnosed. It's extremely resistant to conventional therapy because of this excessive fibrosis. These are the cancer cells, but around the cancer cell, these normal cells are transformed into this fibrotic tissue, which is becoming very dense and very hard for chemotherapy to access these tumors. At the genomic level, we know what kind of mutations are there, but unfortunately, we cannot target them. Because of this, it's predicted to be the second most lethal cancer by 2013. And the reason for this is that for many of the cancer types, actually we have a lot better treatment options now. And the deaths per year are decreasing for almost any other cancers. Now we have better diagnosis, better treatment, and we have cures for some of these cancers. But pancreatic cancer keeps rising, and for the last 40 years, we're still using the same drugs. So one of the research that in the lab is that how can we use this CRISPR screening to figure out what is really forming these tumors and what is allowing us to now use new drugs or drug combinations to kill these cancer cells. So just briefly how we do this, we take a patient tumor, and we are delivering this guide RNAs, but we are delivering thousands and thousands of these guide RNAs into these cells. But each cell is receiving single guide RNA. Now we are putting these tumors back into the mice and letting them to form tumors. And at the end, we are interested in these guys. If our purpose is to identify new novel tumor suppressors, we are interested in these green guys. So these green guys means that initially there was one cell, but when we knock out this green gene, these cells, they proliferate even better, even faster. So these are potentially new novel tumor suppressors. And we have identified novel tumor suppressor that we never knew anything before. For example, one of them is called ISL2 gene. But the more exciting part is we can also use this same strategy to identify combinatorial drug targets. So these are the genes that when we, this is for example this red gene, when we inhibit this red gene normally by itself, the cell, they didn't care too much. They still keep proliferating. But when we add drug, then those red genes, then they disappear from the tumor. So that means that it became lethal. Inhibition of this gene becomes lethal with this specific drug. So it gives us a novel combinatorial drug targets. And we screen for different drugs that are potentially, some of them are currently used in clinics, some of them are potential clinical drugs, and identify completely new set of genes or signaling pathways that we can target to kill these cancer cells. Now, these are the research in the lab, but the CRISPR has many other implications and much global or broader implications for us and human species and also for our ecosystem and our society. But like any other powerful tools, it also brings some ethical questions as well with it. So what can CRISPR do at the global level? In this slide, I put some of the things. Probably this is not a very exhaustive list. For the green parts, it's potentially the good uses of the CRISPR. And these red points are potential ethical issues with it. For agriculture, I will give some specific examples for in animals, plants. It has amazing power to do things that we couldn't do before. At the species level manipulation, this system called gene drive, for example, we can eradicate disease-carrying species. And I will show some examples of this one as well, and can potentially save millions of lives. For example, malaria, because of this disease carried with the mosquito, nearly 750,000 people are dying every year just because of the malaria. And this can potentially get rid of all this mosquito. But I will also highlight some of the potential ethical or moral dilemmas with it as well. Humanized animals for organ transplantation, organ harvesting, for example, and human application. I'll just mention some of the specific examples. In agriculture, the big thing is we know that currently our food supply system is not sustainable. In the next 30 years, there will be additional 2 billion people on Earth, and are currently, because of the climate change, soil exhaustion, loss of farmland, and excessive deforestation, we will not be able to produce as much food as needed for these many things. And CRISPR type of genome editing can potentially become a new resource for us. And this is not genetically modifying organisms, because we have to make this distinction as well. This is using what nature has done in that particular organism and just making small changes to that one. Nature has done it for a number of things. It took for us 10,000 years to convert this into this, but we use CRISPR to do this in just a few years. So we don't have to wait for nature to work for another millions of years on it, or another 10,000 years to do non-naturally select things. CRISPR now allows us to do these kind of things that allows us to increase the yield much greater than what naturally is. And for example, this is a normal beef cattle. It's a very good beef cattle. Not everyone is that fatty. But there is a Belgian blue cattle. This is completely natural. This is not genetically modified. But naturally, there has been some mutation in this thing. And we selected over a long period of time. And now there is a breed of this thing. This one, the only thing that happened is this guy has a normal wild-type gene called myostatin. This is a gene. The protein product of this gene is required to reduce the growth of muscle, basically. This is what the natural function of this gene is. But when a small change, in a small letter change naturally happened, not by humans, in this species, now it gets much more muscular. And this is not only specific to this Belgian blue cattle. There are dogs also with the completely natural mutation in this gene. They're also very muscular. And then there's sheep that are these are non-genetically modified, not by humans, completely natural. And in 1997, you can basically replicate this by research in mice. If you delete this gene in mice, the mice get much bigger and much muscular as well. So nature has done this over a long period of time, and we can now use CRISPR to do exactly this kind of switches in just a year, basically. So this can give us an amazing power to increase the yield for many of these things. This is, for example, this, just to mention this salmon case, I really find it very fascinating. You can change the genetics of this salmon by taking a piece of growth hormone from another salmon species that normally naturally grows larger and put that growth hormone into this salmon. And now this salmon, this genetically engineered salmon, it grows to the market size in half the time that normal salmon does. And it's just because it can grow year round instead of just seasonally growing. And it uses 25% less feed per kilogram gain compared to the wild type salmon. So this is for a cattle, for example, for beef. We have to feed them 8 kilogram of feed to gain 1 kilogram of gain. And for pig is 3 to 1, for chicken is 2 to 1. For normal wild type salmon is 1.2 to 1. But again, this takes twice longer. But for genetically engineered one, it's just 1 to 1. It's still wild type salmon. It just contains a small pieces of DNA from another salmon that now can quickly grow and uses less food. This can reduce the carbon footprint on Earth amazingly, because now we can farm this salmon, which is most of the salmon around us is farmed. Now we can farm these, and so that we don't have to carry them from Chile or from Alaska to our tables. We can grow them in near big cities so that it can reduce the shipments. CRISPR can also save lives and reduce animal suffering. For example, this particular case was a recent news that this pig has been CRISPRed so that it no longer is resistant to this virus that normally kills a lot of these pigs and costs around 600 million a year for these farmers. And when you CRISPR and when you delete these genes, and now they are completely resistant to this virus. Another example that I find really, I was recently getting aware of this process called dehorning. And annually in the United States, 4.5 million of these dairy cows and also 8 million of these beef cattle are dehorned. And the reason is that because of these horns, they can damage each other, and this can lead to infections, and this can lead widespread suffering in these animals, and also they can become very damaging, harmful to the owners as well. So because of this process, they dehorn these cattle, but this dehorning is really inhumane in a way, because you have to literally cut and burn these horns. And now we can use CRISPR to basically, there is a gene that controls this process. We can use CRISPR to modify this gene, and now this is a naturally born animal that is dehorned with the CRISPR system. And this can potentially reduce a lot of suffering in these animals as well. And similarly, we can use CRISPR to grow a longer hair, alter hair color, and this was the salmon case that I produced. We can make eggs less allergic, pigs that are resistant to virus and dehorned. So there's some version of CRISPR that has been used and being used currently to increase the yield or reduce sufferings. There is this also thing called gene drive. Now I found this one of the most powerful applications of this CRISPR technology. It's because now it allows us to eliminate a species on Earth. This is fascinating because every time, all the things that I've described so far, it only goes to that particular individual that we do. If we do this in a pig, that pig becomes resistant to virus, not other pigs. And this trait will not be spread to the population. Or for humans, if I do it on myself, it will only affect me, not anyone else around me. But in this particular case, you modify something in one single guy, and that trait is being spread to the population. And for the sexually growing species, like insects, who are their lifetime is very short, normally in all of us, in sexually reproducing organisms, there are two genes two alleles, basically. And if you make change in one allele, it sticks with this allele, but the other allele is wild type. So if this guy is modified, over the next couple of generations, still has got only a single guy that is modified. But researchers have found that if you find a way to kind of copy this change to other allele, now you can quickly spread this new trait across the species. Because every time there is an allele, it copies itself to the other allele, and now it spreads to the population. So now it is estimated that if we change in 0.1% of the population, if we change this single gene in a mosquito, for example, and release those mosquitoes in the nature, within 6 months or a year, the entire species of this mosquito in that particular region are going to be modified. So what does it mean to modify a mosquito? It's really up to us what we put in this here, because we can make them resistant to we can eliminate the resistance to certain pesticides. We can make them sensitive to a new chemical, because we can design what is going to be carried out in this population. Or this is even more powerful. We can sterilize certain certain sex in this population. For example, we can design this so that all the female mosquitoes are going to be sterile. The mosquitoes are going to still reproduce, but the females for the next generation will not be able to reproduce. So in a way, there will be all male mosquitoes around, and that species will going to collapse at the end of it. So this potentially can cure and save thousands of lives, right? Because 1 million people die due to these mosquito-borne illnesses. Whether it's malaria, dengue virus, Zika viruses, it is all carried with the mosquitoes. But then also, this is amazing power. Now it can carry a lot of, risks as well, because we have never done this before in our history. We have never eliminated a species completely from the face of Earth. And now it gives us to do this one. So when we do this one, and what are the effects in the global ecosystem? We don't know these kind of things. And what happens if this becomes resistant to these things that we changed? And what happens if one mosquito escapes this and start to, for example, spread this thing to other wild types mosquito-related species? We have never done this kind of thing. So there's a lot of kind of risks associated with it as well. But scientists are also developing a lot of kind of the safety checkpoints for this kind of treatments. Bill Gates Foundation, now think they found investing $100 million into this research for malaria to do the malaria research and save people from malaria. Department of Defense invested 200 millions into this kind of research to use this gene drive for various applications. And now finally about the humans. So this is, because we love ourselves, and we are more egoistic in that sense, so we are mostly interested in things that relates to us directly. And the question is now, is CRISPR give us the designer babies or not? So it's funny, because as soon as, of course, the field realized how powerful this thing is, people start asking, where in the world could the first baby be born? And this was in 2015, this article. And part of this article was that, in a lot of parts of the world, there is not a good legislatures or laws and regulations in terms of this kind of genome editing technologies. In the US, Canada and Brazil and other places, there are some laws that bans this kind of applications. But in other places, we either don't know or there are very lax rules about this technology. This article predicted that it was going to be probably either in Japan or in India, because there's a lot of in vitro fertilization going on there and the rules are very lax. However, this wasn't the case, because it came out in China that in last November, the first CRISPR babies are born now by this scientist, Dr. He. Before this one, I wrote this article about this CRISPR ethics, and I guess because of this one, as soon as this research came out, a lot of media outlets and was reaching out to discuss what it means to have a CRISPR babies, even some Chinese TVs, and I assume this is my name. I don't know. So what does it mean to edit human genome? So there are two ways this is done. One is somatic cells. So large fraction of our body is our somatic cells, but there are these few two cell types that we call germ cells. If we change anything to the big part of our body, this is not going to be transmitted to the next generation. So this will be restricted to that individual. If you change in your muscle, in your blood cells, in your brains, anything, it won't be passed to the next generation. However, anything we do to these germ cells will pass to the next generation. So this will be called germline editing, germ cells editing. This will be called somatic cells editing. So this one, I will mention how we can do this one. But the only way germline currently, the germline editing can work is if we use CRISPR to change things at the very beginning of embryonic development, basically in the zygote. As soon as the sperm and egg fuse together, we can add CRISPR to this one and make changes at this stage. This is the only way it can happen. Of course, this has a huge ethical concerns, moral dilemmas about it. Should we do it? Should we not do it? I will come to this one. But for somatic cell genome editing, this has this can potentially cure a lot of genetic diseases, because we can now correct these disease-causing mutations from an individual, take the cells from individuals and correct them. There are two ways we can do it. One is for certain cell types, for example, blood malignancies, we can take the blood out and we can culture these cells. We can add the genome CRISPR and correct these cells and gives these cells back to patient. And this is completely now edited, and we can correct a lot of genetic diseases like this. And FDA just approved the clinical trial for cancer. A lot of blood malignancies such as sickle cell diseases, beta thalassemia, or HIV can be potentially cured this way, because we can easily take the cells out of patient. We can ex vivo grow these cells. We can modify them and give the patient this corrected cell. This is the easy route to do these kind of things. A bit harder way is to do in vivo gene editing, because a lot of cells we cannot take it out. For example, in case of muscular dystrophy or cystic fibrosis, a lot of muscle cells, for example, is affected by muscular dystrophy. We cannot just take muscle cells out of the patient and correct them. So we have to be able to deliver this powerful tool into the individual, into these local sites. Of course, this is much challenging than this approach. But for certain diseases, it affects particular organ much more than other organs. For example, in cystic fibrosis, it is the lungs that are affected. So if you could cure only 10% of the lung cells, change this genetic modification into normal, and you basically eliminate the disease. You don't have to cure 100% of the lung cells to cure cystic fibrosis. 10% of this is enough. And this has potential treatment and cure really for a lot of genetic diseases as well. But when it comes to germline editing or embryo editing, it's a different situation, because now we are literally changing or evolving ourselves into a completely different route that we have never done before. However, I should mention that there are genetically modified people among us, and for the last 20 years. And this is interesting, because we thought this comes with the CRISPR, but this is actually before CRISPR. In 1997, an IVF clinic in New Jersey published this paper. The title is, it has a lot of kind of jargony things: birth of an infant after transfer of anucleate donor oocyte cytoplasm into recipient eggs. Basically, what they did is that a new baby was born from 3 parents. So how does this happen? Is that there is this mitochondrial replacement therapy. This is for this genetic diseases because of this defects in the mitochondria. So mitochondria is a powerhouse in our cells, and the mitochondrial has its own DNA, separate from our own genomic DNA. And this DNA comes only from mother. So if mother has a defect in mitochondria, all the babies will carry out this defect. What they did, basically, they got the mother, because these two individuals, they want to have a baby, but this mother's mitochondria is defect. So what they did, they take the nucleus of this mother, and they take the cell from another mother, they take its nucleus out, and put this nucleus into the other cell. And now they can fertilize this make a zygote and implant back into this mother and have a baby. Now that genetic mutation or genetic disease has been corrected. However, now this new baby has a genetic information from its original mother and father, but also the genetic information from the third mother is still present in the mitochondria. So this whole next generations will be germline modified, basically. So when this paper was published, of course, the FDA and the regulatory mechanism in the US were not happy. So they inserted this new clause into the budget that said research in which a human embryo is intentionally created or modified to include a heritable genetic information modification. The FDA does not even allow discussing this kind of research now. So, but what happened? So, and in the UK, they had a large public discussion about it, and in 2015 they passed a law. Now it is doable in the UK, but in the US we still can't do this in vitro fertilization. It's funny because in 2016 a couple wanted to do this one with this doctor who originally was doing this research, but they couldn't do it in the US, so they just paid the doctor. They took him to Mexico and did this mitochondrial replacement therapy in Mexico and fly back to the US. So once these things are doable, it's very hard to contain this kind of things. But of course, now with the CRISPR, it's doing much more than that. So with the CRISPR, there are of course some moral or ethical questions that are related to CRISPR, and the others are, even before CRISPR, we were discussing these things. Our moral values are, dictated by religion and by our culture. So those two things can really define what is ethical, what is not ethical for certain things. For embryonic research, for example, we have always been discussing this in previous to CRISPR technology as well. What is really embryo? This picture didn't show up. When a sperm and egg fertilize and that is one cell, is this really considered, should it be considered embryo, or is it just a single cell? For example, in Christianity, the religion said it is an embryo, but in Judaism and in Islam, that stage is not considered an embryo. So now you can consider how this cultural or religious values dictate what is ethical, or what is moral, or what is not. So again, we're going to continue discussing these things. And when does an embryo really start having its own dignity or its personhood? Is it at a single stage? Is it when the heart beats? And is it, when is this stage? Science cannot solve this one, basically. This is not an area of a science. This is completely needs to be discussed, and our religious values, cultural values will define this one. But there are also some ethical concerns that starts with the CRISPR when it comes to germline editing, because now the question is, why are we doing this? Like, why are we doing the germline editing, changing the DNA of a next generation? Because anything we do is going to pass on to all the future generations from there on. So why are we doing this? Are we doing this to cure a disease, for a treatment purpose, or are we doing it for enhancement? And I'll mention some of the difference between these things. One is, you can say, if you don't do this one, all the next things we're going to suffer. So we have to do this to cure this disease. But it's a slippery slope. And who is responsible for the risks? Because biology is complex, and I will come to that point as well, that we can't really know for sure what happens when we delete a gene from a human, because we have never done these experiments before. And who has the right to consent? You know, should parents' consent be enough for embryo? You know, you can say, but the parents, they can be responsible for that particular embryo, but what if it's future generations? Because currently, when a baby is sick, parents can give consent to do a lot of treatments on the baby. But usually, typically, with the typical treatment in current hospitals, for example, the parents give a consent because they are stuck with that risk with that baby throughout their life. But in this case, it's not only with that baby, but also the future generation. So now, who should give the consent, and who should be responsible if anything goes wrong? Should it be parents, doctors? Who is going to be responsible, and who will have access to this? This is more global societal issue. Is this going to be accessible to everyone, and how expensive it is? Because this has to go through IVF, and IVF currently is $20,000 to $30,000 in the US, for example. And a lot of people, of course, won't have this accessibility or have the power to access this. Will this create this social injustice? When, of course, the committee in 2017 met, and there is currently a moratorium on germline genome editing, which is basically all the scientists decided that we should not do the germline editing until we further discuss these issues. But declaring a moratorium doesn't mean that you can enforce every other scientist on Earth, because as in the case of Chinese scientists, some rogue scientists can still go on and do these things. But they concluded that if there is, we should never try to do the germline editing unless for the treatment purposes. And in the treatment case also, or the cure case, there shouldn't be any other alternative options. We have to know the conditions of this disease, and we have to know which gene is causing this, and it needs to be only converting this disease version into a normal version, not enhancements. But of course, we don't have a regulatory body across the world that can enforce these regulations. You know, it's an interesting dilemma, because if CRISPR can cure a genetic disease, and now we have this technology, should we use it or not? That's a main question, because some people say that if one argument is that we should not do it because this is not natural, this is not how life worked on Earth. But another alternative argument is that, well, this is the most humane thing to do, because if we don't do it, those people are going to die, and we may be responsible for their death. So now you can see how these two things kind of may contradict to each other, these two kind of moral values. And some, bioethicists say, intentionally refraining from saving a life is be morally responsible for their deaths. And now the next thing is, the genetic treatment generally, a lot of, there's some sort of agreement on it that, it can potentially and should be used to cure genetic diseases and reduce suffering and save lives. But as soon as you start to go to that route, now it's a slippery slope, because now we come back to the enhancements as well. So in this case of enhancement, we are basically giving a new trait or enhancing an existing trait to that individual. So for example, for cardiovascular diseases, we know like certain genes are very high risk for developing cardiovascular disease and increased cholesterol. And of course, if you don't do it, those people will die much earlier. You can say that this is a disease situation, we should correct it. But you can also, some other people will say, well, mine is around normal, whoever decides it's normal, but I want to further reduce my cholesterol level, right? Even be healthier. Now this can go into the enhancement. And as soon as we started this one, it kind of creates this fear of eugenics. The eugenic movement, as you know, was this idea or this practice that certain traits in a population should be selected, and other with the selected breeding among humans, and some other traits a life that is not worth a life should be discriminated or should be get rid of. And this, in the United States, this research was very active in early 90s. And with, as you know, with the Nazis, Germany, this came to a new level when the government enforces eugenic movements and forced sterilization of vast number of people and killed them. And in the United States, it also happened that certain population became disadvantaged. They forced sterilization in the United States. And unfortunately, UVA was one of the active sites of eugenic research. And the building that my lab was used to call Jordan Hall. And Dr. Jordan was one of the forefront researchers of this eugenics movement. And recently, realizing all this dark history of this eugenics movement, it has been renamed. So, of course, who will have access to this technology, and will it create a social inequality? I think currently we are banning this kind of things, but the Pandora's box is open. We will see in our generation probably edited gene editing babies, even for the enhancements, because an argument is, for example, they say we enhance ourselves in all sorts of ways, right? For example, we drive nice cars, and this is a major enhancement for us. It used to take us, years to go from one place to another place, and now we can just fly there, or we can just drive there. Or we wear eyeglasses, and we wear shoes. This is all sorts of enhancement. This may be just one of them. And the alternative, or the argument against this is that, well, if you do this one, then you create this injustice in society, and certain population will have advantage, the others won't. Yes, but they may say, or there's that, well, there is already a lot of inequality in society, right? Rich parents can enhance their kids through specialized education, specialized classes, tutorings, etc., etc. So they are already enhancing this certain population, and this could be just one of this kind of enhancements. So the Pandora's box is open, but we as a society, I think, needs to discuss this thing. Typically the scientist's view on CRISPR is, there's a lot of research application. The ethics, we tend to think about it as in terms of sensitivity and specificity. Does it have off-target effects or not? This is a scientist's view of kind of CRISPR. But it's not about whether the CRISPR has some off-target effects or has some side effects. Even if it's a picture-perfect technology, it's still these moral and ethical questions are going to remain there. So we need to have a much bigger discussion on CRISPR, not only on scientists, but ethicists, social scientists, legal scholars, religious thinkers, social classes, and probably many other classes in the society needs to come together and have this public discussion. And it is likely that some of these things that we discuss right now will be gone in next decade or so. Because think about the in vitro fertilization. When in this Louis baby was born in July 25, 1978. You should see the headlines of the newspapers back then, that this is a monstrous, we should never do this, and super babies born, etc., etc. Because these parents naturally were not supposed to have babies. But we humans, we changed the course, and now 8 million in vitro fertilization babies are born now. So 8 million of them, and the society completely accepted this right now, right? 8 million is the size of the whole state of Virginia, basically. So we have a lot of IVF babies born, and it could be that the CRISPR could be one of those things in the next few decades. So will that ever be a designer babies? This is, if we say that CRISPR can designer babies, I think this will be a very narrow point of view, because really we don't really know what makes us human. Genetic is the major constituent of this definition, but genetic itself is not enough, because we have, in addition to our genome, we have our epigenome that I described. How is this genetic information regulated? But in addition to this one, our microbiome, our virome, are other genomic information, are the other information that we carry in each of us. The microbiome from one individual to another one is completely different. Even if the genetic information is completely the same, if the microbiome or virome is changing, that person, the character will be different. And we carry many more microbes in our body than the total number of our cells. So you can imagine how much additional information we carry with each of our cells. So I want to finish with this thing, that will we ever get a designer babies? I think this quote from Francis Collins, the current NIH director. This quote was when the human genome was being read, that there was the same discussion, will the genomic information now allow us to design babies? And he said that we'll get closer to perfect kids from caring families and good education than we will ever get with the genetics. And this is probably true that for the CRISPR as well, that genetic information is one thing, it's a major thing, but probably it's not enough to design a certain character. So with this, I would like to thank you for listening, and I would like to thank the people in the lab who are involved in this, and thank collaborators and the funding resources as well. So I'll be happy now to take any questions that you might have. So I know that the ethical is a big thing. I probably raise more questions than answer some of these ethical questions in your mind, but I just want you to be aware of these things. So thank you very much.
Does anyone have any questions?
So it's us. We can ask any questions if you have.
So I had a kind of a technical question. So when I read the paper that came out with the Chinese paper, the Chinese CRISPR babies, I understood that he had edited a gene that allows or prevents HIV infection,
Yes.
which to me seems like an enhancement more than a prevention of or curing of a disease.
Yes.
But then there was also some technical discussion about that he had not actually designed his target RNA molecules correctly. Or can you talk a little bit about that, or what your sense of his technical expertise was as well?
Yes. So let me. Okay, this is what he did actually. So he designed his guide RNA to delete this gene basically, and this is in his mind is a doorway where the HIV virus gets into our cells. So when there is in some populations, human population, they are naturally knocked out again for this gene. So they are resistant to certain HIV viruses, certain types of HIV viruses, not all of them. So he designed to knock out this in this embryonic stages. So there were a couple of problems with this. One thing was it medically needed? Because there was alternative ways to completely cure these babies with HIV, because their father was HIV, but their mother was normal. So there are ways to basically wash the sperms and so that it won't carry the virus and it won't cause the next generation HIV. So and the other thing what he did is, so he's not a CRISPR person. He has never published CRISPR papers before. He is a physicist who now has access to these things, and he had the expertise with the IVF technology, and he is carried out because it is easy to do these kind of experiments. But one of the challenging things that he couldn't quite answer was the baby when the embryo is developing, it starts from a single cell and divides into 2 cells and 4 cells. Some of these babies were one of the baby was mosaic, which means that some of the cells in the body was edited or knocked out for this gene. The other cells were not edited. So that baby is completely still sensitive to the HIV infection. So it won't be cured. The other baby, we still don't know exactly whether it's cured or not. And the other problem with this particular gene is doing this one is that it can only prevent the infection with certain types of HIV virus, but others HIV virus can still infect. And more importantly, it can even make these babies more sensitive to, for example, West Nile virus. The HIV cannot go, but these babies can be more sensitive to even some additional viruses. So there's a lot of problems with it. And then studying the off-target effect were not properly studied. So a lot of scientists call this, unethical, dangerous, and irresponsible act of doing science and trying to do the genome editing in babies. I don't know if this was the answer to your question, but
I have one question about the enhancement theory of CRISPR-Cas9. So when you describe the animals, like they have the horns, when we, silence the genes, they don't have horns, or we can add more hair to the animal or less hair to the animal, or the human being, we can change what based on human being they don't want, we can change it, right? Yeah. But what we have is the animal has a horn because it's an adaptation mechanism. It's naturally there because it helps it. Or the mosquito has this something that impacts negatively us, but it may help the mosquito to survive. Those are the adaptation mechanisms that we have those features, or the animals have those features. So if we change them, I'm talking about the next 5 decades, like later, like maybe 100 years later, when we use the CRISPR, which means we may gonna get, instead of getting an enhancement, maybe we or the animal's gonna be maybe more vulnerable to different type of disease, because losing one piece that's really essential for you being survive, but maybe we don't know, can open a new era of disease maybe. Yeah. It can be possible. It's a kind of like, we don't know actually, we are changing. Yes. But it has been there for essentially for
reason, right?
Survive you, in the nature. Yeah.
No, I completely agree with you. These things naturally have evolved that way, and we think it must have some function to it. Yeah. And if it changes, what happens? We exactly don't know. In certain cases, what we know is that there are natural mutations. So, for example, there's a Belgian blue cattle things. There are some species have naturally evolved that way.
But that's needed, right, for the species to survive. That's why they had the mutation based on the region maybe they leave, right?
We can imagine why horn can be a very powerful thing for animals in the wild, right? Because it is the only thing that they can fight with. Yeah. But if for a cattle grown in a small, closed environment, we can also know how this horn can now become a disadvantage for that animal and also for that species. For this kind of contained environment, dehorning, can be advantageous for that species, because evolution worked over billions of years is not really for today's environment, right? We have, as a human species, we changed our environment completely. This environment wasn't there 50 years ago, 100 years ago, 1,000 years ago. But this the evolution of horns happened over a period of millions and millions of years. But you can say that evolution is kind of blind to the future, right? Evolution won't know what the future looks like, and the things will change, will evolve with the future potential. But you are right. We are changing something that we don't know what's going to happen, and
we are actually on the way that redefining the Darwinian natural selection.
Exactly.
But we don't know
which direction is going to go. And the other thing is, we think that evolution has perfected things, right? But the other argument is that we know that many species go extinct throughout evolution, right? So evolution is not trying to perfect things. It's just selecting whatever trait is available at that time with those conditions. So it is an alternative approach that things are not perfect as they are as a species, and they will keep continuing to evolve whatever pressure is on those species at that particular time.
We may need assessment forms before we get the genome editing, maybe based on where we are, who we are, what is necessary, and then they're going to make the modification.
Ideally, yes, but when we come to the genetic editing, a lot of things that we don't know. Even for certain traits that we think should be helpful, that, for example, with the CCR5, right? We can say this is, there are some people who are knockout for this gene and they are resistant to virus. And we can say, wow, this is, if we delete this one, we will be becoming resistant to HIV. Yes, but then you are becoming more sensitive to West Nile virus, for example. So it's really difficult to predict why certain genes have been, still have been evolved that way. Like for some of the genetic diseases, for example, it's really peculiar why after millions of years of evolution, there are still certain traits, genetic diseases are still contained in human evolution. Because you can imagine that those should have been selected out, but somehow they are still being carried out throughout evolution. So one possible explanation is that, yes, that gene, for example, for Huntington disease, at the later stages of life, it is very devastating. But evolution doesn't really care about late stages of life. Evolution cares about reproduction. And during the early stages of life, those Huntington people, they're actually quite smart. And they are active, they are very smart, and they have enhanced memory. So you can see now how a gene can cause a genetic disease, Huntington, devastating genetic disease, but at the same time, at the early stages of life, can have a very beneficial effect. And that's why this trait is still being contained in our society. And if we try to edit that Huntington gene, whether these people will be, have less memory or less reproductive, we don't know.
Anyone else?
So kind of overall, looking at I guess this field across the board, is there kind of a consensus of when it comes to the risks versus the benefit? Is there kind of a idea of should we just kind of deal with the risk while we can reap the benefits, or is it kind of more along the lines of we kind of should kind of hold off in general?
The current thing is actually there are two kind of school of thought. One is that for a lot of these genes, for certain genetic diseases, we know the benefits. If we could edit this gene for cystic fibrosis, for cancer treatment, there's tremendous benefits to it. But the other school of thought is that we don't know. So if we don't know, we should wait and do more research, basically, on this kind of things. But then the alternative school of thought is that we do a lot of treatments without really considering too much about the side effects of it. For example, whenever you have a cancer, you do radiation treatment, right? Is one of the treatment for cancer. But when you do radiation, you basically mutate tons places in the genome. But we still accept this as a perfectly fine way of treating a disease because we think this has benefits that outweighs the risk. Compared to the radiation treatment, for example, the total number of mutations, off-target mutation may be caused by CRISPR is probably a fraction of the radiation mutations. But we are still worried way more about CRISPR than the radiation. And when the school of thought, one school of thought is that why are we holding CRISPR to a much higher standards while we are using chemotherapy or radiation therapy for a lot of these kind of diseases? So one says that if we wait, we have to eventually do these kind of things. By just waiting, it's not going to solve our issues. But how are we going to do it? Eventually, we have to test this in embryos. But then all the moral, ethical dilemma starts. And what is embryo? What stages of things we should be doing, etc.
Thank you so much. That was
Thank you very much.